Good morning and good afternoon, everyone, and welcome to Fierce Innovation Day. I'm Julien Meissonnier, and I will be your moderator today. I currently serve as a board member at Prolific Machine and senior advisor to life science innovators, service provider, and investor, helping them with their strategy, technology, and commercialization across all therapeutic modalities. So, Prolific Machine is one of the most exciting technology platforms I've seen in biologics manufacturing in a very long time. It's exactly the kind of breakthrough innovation that belongs at Fierce Innovation Day because it has the potential to fundamentally change how we control biology during biomanufacturing. So today is not meant to be a deep scientific lecture. Instead, we want to focus on four things: what problems this technology can solve, why it matters for innovators, how it works at a practical level, and how companies developing products can potentially partner with Prolific to improve development and manufacturing outcomes. So if you want about Prolific, it's an impressive company based in the Bay Area. It has a great lab, pilot plant, and a team of more than 50 scientists and engineers working on the technologies and solving some of the most complex challenges of today's biomanufacturing. What also makes this tech especially exciting to me, and we're going to discuss it today, is that it can apply across therapeutic protein, mAb, multi and bispecific, complex fusions, and even biosimilars. So having spent nearly 2 decades helping multiple companies bring biologics into the clinics and ultimately to the market during my time at Catalent, I genuinely believe this team is bringing something unique to the industry. So with that, I'm very pleased to welcome my friend Max Hörner, heading the Optogenetics platform at Prolific Machine. And Max, great to have you with us. Right?
Yeah,
so,
good to have you.
And Max, first question. So one of the things that immediately caught my attention was the productivity data you generated lately. Right? The company recently released breaking over the 20 gram per liter titer on a very complex mAb, and with a path to reach even higher titer and maybe 30 gram per liter. Everybody could be hopeful about that. And honestly, in fed batch, those are numbers many people would not have expected to see, starting with me. I would not have been expected to see that. So maybe starting from a high level, could you please explain us in simple words what is Prolific doing differently and how does optogenetics actually work in this context?
Yeah, so I think it's a good start. So to look at our platform, I think the main difference what we are doing is that we are directly controlling the cells with light, in particular the gene expression of the cells with light. And this is fundamentally different to what the industry has done so far, kind of where you're mostly kind of constitutive promoters like CMV promoters were used, and the cells were expressing all the time, but at the end, kind of very indirect stimuli like temperature shifts for production were used to control the cells. And kind of at Prolific, we really kind of believe light is the best way to control biology, and the direct kind of intervention, kind of the direct interaction with the cell is giving us that fundamentally kind of new control lever. So we cannot only turn gene expression on and off during the production rate, during the production run. And we really can precisely tune the expression level to the optimal level throughout the production. And also, when we turn the light off during production, we can revert the expression. And this is fundamentally different to chemical inducers and brings us that direct control to the cells. And in addition, I think what's very exciting is that we can use light of different colors to control multiple cellular processes in a cell fully orthogonally at a given time, just by illuminating with different light colors. And so how are we doing that? So we're making use of optogenetics. And in the field of optogenetics, we are transferring the genetic information from photoreceptors, from light-sensitive organisms, into our target cells, kind of rewiring them with the cellular machinery, and then have direct control of the cellular function. And we transfer our photo switches into all kind of mammalian cells, but mostly suspension CHO cells, suspension HEK cells, and control their gene expression with light. And so I'm now working for the last 15 years in optogenetics, kind of mostly in the academic context. So I joined Prolific 4 years ago. Before that, I was heading a research group at the University of Freiburg in Germany, also focusing on optogenetic control of mammalian cells. And kind of while in academia, light control gene expression system have already been shown, but kind of I know those systems kind of very well. And I would consider them more as a kind of a proof of concept study, kind of developed on a high fold change, but not necessarily on high kind of transcriptional output. And those systems don't have the performance required for biomanufacturing. And this is where we spend kind of significant resources at Prolific to bring those academic system towards industry performance, meaning they are able to produce proteins in the gram per liter range. And this library of our kind of own developed light-inducible, proprietary light-inducible gene expression is really kind of at the foundation of our technology.
Oh, great, Max, thanks. That idea of directly controlling cell biology is fascinating to me because historically the industry has mostly over the past 2 decades tried to influence cells indirectly, right? To media, feeds, process conditions, or even genetic engineering approach. But at the end of the day, you put the cell in the most healthy conditions and they need to crank the stuff in the bioreactor. So what is impressive here is that the platform still seems very compatible with existing fed batch infrastructure and even continuous. So how easy is it for company to integrate this into current manufacturing environments? Are we talking about a complete change in infrastructure, or is it just something that is plug and play and can work relatively seamlessly within today's biomanufacturing and bioprocessing operation setup?
Yeah. So I think a great advantage of our technology is that it's really compatible with existing infrastructure, kind of both on kind of cellular level, but also kind of on the bioreactor level. So maybe let's start first with bioreactors, fed batch bioreactors. So at Prolific, we have a hardware team and we are developing modular illuminators that can be kind of added in a plug and play manner to already existing bioreactors. And I really want to be clear here, we are not developing any new bioreactors that are just modular add-ons for already kind of existing bioreactors. And in the case of single-use back reactors, we deliver the light through the transparent back just by placing our modular LED modules outside of the back. And the great thing is we're here not breaching the sterility barrier. We're just there's a minimal kind of intervention kind of with the already existing system. We're just delivering the light through the back. And this makes it compatible kind of with all of the bioreactor systems from all major vendors like Sartorius, Cytiva, Thermo Fisher. Kind of it's really a modular add-on.
Mm-hmm.
And besides the besides the single-use back reactors, we have also developed illumination systems for stainless steel bioreactors. So these are external illuminators that are flow-through illuminators similar to kind of a perfusion system where we are pumping the cells through a transparent illumination loop where we are delivering the light to the cells. And to date, we have now scaled the technology at Prolific in our pilot plant up to a 50-liter single-use back reactor and a 200-liter stainless steel bioreactor. And we have developed all kind of the models that were able to translate our light patterns from a small-scale 24D full plate format up to kind of those large bioreactors. So kind of the light delivery is something that we can model, and it is really predictable to the larger scales. And we have done those runs kind of with the intention that we are de-risking and developing everything to scale towards larger scale. So currently we're working towards scaling the technology towards 2,000-liter single-use back reactors. And we also kind of have in our pipeline plans to scale to larger stainless steel bioreactors. And I think ultimately we are able to address all commercially relevant scales with our technology.
No, that's great, Max. Yeah, no, I'm glad to hear that you got everything compatible to the cell line development platforms. You got the illuminator to go there, and I mean, you're even going to 200 liters, so you can do preclinical batches manufacture at Prolific, and I know you did. And now adding to 2,000 liter subs with with a manufacturing partner is going to be. I mean, the speed at which you've been doing that is pretty spectacular, right? So building on that, where do you think this technology creates the most value today? Are there any particular class of therapeutic protein or development challenges where Prolific's platform really stands out?
Yeah.
So in a nutshell, where are you seeing the strongest fit for the audience?
Yeah, I think the most obvious fit of our platform is the expression of very challenging, difficult to express proteins, such as highly complex antibodies, biospecifics, fusion proteins, Fc fusions, cytokine fusions. Because the reason for that is that protein production and cell proliferation are always competing for the same cellular resources. And you always have to make a trade-off between an acceptable growth rate and a high specific productivity. And with our light-inducible systems, we're able to get the best of both worlds. We don't have to make that trade-off. We can grow the cells rapidly in the uninduced state to high cell densities, and then turn on protein production with light, and thereby are able to get much higher titers and kind of specific productivities. And we typically, kind of based on our experience for conventional maps, we have shown for multiple of them that we are able to double the titers compared to the conventional process. And in a current partnership with a partner, they were interested in kind of exploring our platform for 2 of their maps, because they want to really bring kind of map titers kind of to a new level. And we were working with them on a easy to express map and a difficult to produce map. And while I think similar kind of to our kind of in-house experience, we were able to double the titer of the easy to express map. I think what's even more exciting is that for the difficult to produce map, we were having a kind of a 5-fold boost in titer compared to our matched CMV control. We are bringing the titers in also for those challenging molecules to the same level of those easier to express molecules. And I think this is really where I see one key value of the platform. It's not just about boosting titers, but it's really bringing also titers for challenging molecules kind of to a range where they are commercially viable, and kind of thereby significantly reducing the developability risk of those new molecules. And besides just kind of separating the growth and production phase, I think a very exciting application of our technology are biospecifics and other complex multi-chain proteins. And for those molecule classes, kind of the key challenge the industry currently faces is that you need to precisely titrate the expression level of the different chains of the different subunits to maximize the correct assembly. And this is typically done currently on the genetic level. So with a lot of different genetic constructs, maybe making use of semi-targeted integration to really kind of bring in kind of diversity and experimentally identifying the pools that have the kind of optimal stoichiometry between the different chains. And this is a very kind of screening process, linked with with a lot of kind of resources. And our approach differs in that way, that we work with a standardized set of vectors, and then we have some chains under constitutive control and some chains under light control. And then we can use light to really titrate the expression level of the different chains to each other. And thereby we are able to kind of accelerate the development timeline, because we don't have to go through that manual screening process. We can save at least a month in that development timeline, and then use light to precisely adjust the expression level. And we started recently a collaboration with Radiant. They have a very complex multi-body platform with a lot of different subunits, and I think they're kind of our technology kind of is very well suited on really optimizing, balancing the different kind of ratio of the different subunits. And yeah, I'm really looking forward on being able to share data of that collaboration soon. And I think maybe the last application I think I'm really excited about is controlling the PTM profile of proteins with light. I think that's very exciting for biosimilars. And I think as you all kind of know, biosimilars, the key challenge is to meet all the critical quality attributes of the innovator drug. And regarding kind of those critical quality attributes, matching the PTM profile is typically one of the key challenges. And with our platform, we can control the expression of those enzymes that are doing those post-translational modifications with light. So, and this means if we can tune the expression level of those enzymes, we can tune the level of the PTMs of, for example, antibodies. And we have recently demonstrated that by showing kind of precise control of galactosylation and sialylation with light, and kind of thereby bringing kind of a fundamentally new lever to control post-translational modifications of proteins.
Yeah, indeed, better control over cell biology could potentially improve not only productivity, can improve productivity, but also product quality, subunit, and PTM. So, and ultimately, the probability of clinical and commercial success. And as many of us know in the industry, those are absolutely critical, especially for complex biologics and even biosimilar, as you name it. So therefore, in a couple of minutes, right, if I'm an innovator, and I know you work with a lot of company right now and customer and partners, right, recently with a biotech that was stuck with low titer in phase 2. And if I'm an innovator developing a novel biologics, whether that's a monoclonal antibody, an ADC, multispecific, or even another complex protein fusions, you've been able to express IgG, cytokine and things like that. How could I engage with Prolific? And similarly for biosimilar developer trying to maximize their chance to get pharma equivalents, how can partnering go with Prolific? So could you give us a practical way of how to work with Prolific?
Yeah. So kind of overall, kind of we are very flexible in the way how we are interacting with our partners. And we can either start kind of the development with the cell lines that we have developed in-house. I think a huge advantage is we can also integrate our optogenetic system into the cell line of our partners. And that has the unique advantage that the partner can really kind of stick and kind of profit from all the historical knowledge and protocols they have kind of accumulated over time with their cell line. And so typically kind of for smaller biotechs that not necessarily kind of have already kind of their own CHO platform, kind of we are doing the cell line development for them, kind of using our established CHO cell lines that are optogenetically enabled. And kind of you previously mentioned kind of our collaboration with a biotech that is currently in clinical phase 2, and I think that is really a good example on showing where our technology can help us. Kind of they have really exciting clinical phase 2 data, but the titers are pretty low. They are in a 0.5 gram per liter range. It's really a challenging molecule, and the costs kind of associated with the production are really kind of prohibitive on going to clinical phase 3 and in particular for commercial manufacturing. So they are looking for solutions on bringing up the titers, and they worked already with more than 4 different of the leading CDMOs, and they all hit a titer ceiling in that range of 0.5 gram per liter. And we used our platform for that, kind of the inducible expression, and we were already now able to double the titer, and that makes a huge difference, kind of in particular at kind of in those in kind of when you are at
Awesome.
lower overall titers. And kind of we are currently working with GMP manufacturing partners, so that they are doing the clinical manufacturing, or can do the clinical manufacturing of that molecule in the future. And
yeah, thanks, Max. Yeah, really exciting discussion, Max, and thanks for clarifying. You do cell line development for them, and then you can support them up to preclinical batch manufacture and carry on to GMP manufacture with manufacturing partner, right?
yeah.
So thank you. Really exciting discussion. I really believe Prolific will reshape bioprocessing and unlock a new wave of biologics innovation. There's no doubt. So thank you as well to everyone joining us today for Fierce Innovation Day. We really appreciate your time and interest in Prolific Machine. Thanks for your attention.
Thank you.